Hello, everybody. Welcome to this webinar organized by EURO-NMD in collaboration with the Rare Neurological Disorders Reference Network and the European Academy of Neurology. Today, we are going to have a webinar on the new Limb-Girdle Muscular Dystrophy classification by Professor Volker Straub. Harold Macmillan, Professor of Medicine and Professor of Neuromuscular Genetics, also Newcastle University Deputy Dean and Director of the Institute of Translational and Clinical Research of the Newcastle University, and he leads the John Walton Muscular Dystrophy Research Center for Clinical Genetics. He trained as a pediatric neurologist and did a PhD in Duchenne muscular dystrophy and worked as a postgraduate with Kevin Campbell's laboratory in Iowa on Limb-Girdle muscular dystrophies. He was a co-founder of Untitled, a very known alliance that was launched with the EU FP6 funded network program, and is a long-standing member of Untitled TACT committee, which offers guidance on translation and development path of therapeutic programs in rare neuromuscular diseases. His current research involves application of muscle imaging, use of severe fetch and mouse models, next generation sequencing and other omic technologies for the characterization of primary neuromuscular disorders. Before passing it over to Volker Straub, I want to just remind you that you can put questions through the Q&A tool, and you can chat between yourselves through the chat tool. Volker, it's a pleasure that you've accepted to give this webinar, and the floor is yours. Thank you.
Thank you very much, Antonio. I'd also like to thank EURO-NMD actually for this opportunity, and the muscle group within EURO-NMD for accepting this proposal. Now, I will not talk about limb-girdle muscular dystrophies. I will really talk about the classification. Of course, I will elaborate a bit what limb-girdle muscular dystrophies are in the history, but nevertheless, the talk will really be around the classification. For this talk, I don't really have a disclosure statement because it's about classification. Nevertheless, industry is actually quite interested, and I'll come to this later, to also have some consensus about classification. I'll first give you a bit of historic background, because limb-girdle muscular dystrophies have a long standing relation to the center here in Newcastle. And it was John Walton from Newcastle and his mentor, Frederick Nattrass, who actually first suggested the term limb-girdle muscular dystrophy. And this was in his seminal paper from Brain in 1954. Those who haven't read this paper, it is really worth reading. It is, I think, 68 pages, but nevertheless really fantastic, because he summarizes everything what we know about muscle diseases in that paper. And he said, well, leaving aside certain clinical characteristics, which may be added later, we feel that the cardinal features of the limb-girdle muscular dystrophies are onset usually late in the first or in the second or third decade, but sometimes in middle age. I always thought that's the same, but anyway. Commencement of muscular weakness in either the shoulder or pelvic girdle, transmission usually via an autosomal recessive gene, usually, so they already knew that there were dominant ones as well, and a relatively slow course, which nevertheless leads to severe disablement and often death before the normal age. So it's actually a really good clinical classification. Most of this actually holds on to limb-girdle muscular dystrophies these days as well. Now, it took about 40 years before the first gene was actually characterized. And in 1994, Alan Emery and the ENMC had just been established 2 years before, published a booklet about the diagnostic criteria for neuromuscular disorders. And it was really great at that time. And there was one chapter in it that was called Limb-Girdle Muscular Dystrophy. And there was a young clinical geneticist called Kate Bushby, who actually wrote about the diagnostic criteria. And so it was very brief. So this is page 25 here. And so it's about the clinical, laboratory, and DNA studies. And clinically, it was basically, weakness that is mainly proximal, and then, it's progressive. And then laboratory, the recessive ones have a high CK, the dominant not necessarily. And then in terms of DNA studies in 1994, when this was published, there were 2 genetic loci, one on chromosome 15 and one on chromosome 2. 15 locus was calpain 3 and the 2p one was dysferlin. There was also one biomarker in 1994, deficiency of the 50-KD dystrophin-related protein. Now, later on, that protein was called adenylin and is now called alpha-sarcoglycan. And the conclusion was that actually, in order to diagnose a patient with limb-girdle muscular dystrophy, you need to have an absence of or abnormality of the expression of this 50-KD glycoprotein. That was the only biomarker. Now, Katie, together with Jackie Beckman, also came up with the first classification system or nomenclature for limb-girdle muscular dystrophies, and that was at a ENMC workshop that was published in 1995. And you can see here Alan Emery, and then you see Jackie Beckman next to him, and Fernando Tomé, and next to Professor Hausmanova, you can see Katie Bushby. You can't really see her because she's just chatting away with Louise Anderson, also known as Louise Nicholson, here from Newcastle. There are a few familiar faces. You see Marianne de Visser, the chair of our muscle group within EURO-NMD. You also see here young Andoni Odis Barrera. There's Thomas Voigt. So quite a number of eminent neuromuscular clinicians, several of which had big families with limb-girdle muscular dystrophies as well. And the classification system that they came up with was based on the mode of inheritance and then a letter. And so the mode of inheritance was the recessive ones are going to be called LGMD2 and the dominant ones LGMD1. And then you add a letter in the order of their first characterization. And the reference for this characterization wasn't the gene, and it wasn't the protein, but it was actually the genetic locus, because in 1991, Jackie Beckman and his group were able to localize the first LGMD gene to chromosome 15 by a linkage analysis. And so was the group around Marcy Spear, that there was linkage of the first dominant form to chromosome 5q. Now, these genes were only identified, several years later. Nevertheless, that's the reason why these are 2A and 1A. The first gene that was actually identified for limb-girdle muscular dystrophy was by Kevin Campbell's lab, and this was Steve Roberts, who was a postdoc at that time in Kevin's lab. And they identified a missense mutation in what they called the adenylin gene, and it was linked to an autosomal recessive muscular dystrophy. So it was called 2D, despite the fact that this was the first gene, because there were other genetic loci for actually some of the other sarcoglycans as well, and then for dysferlin, that were identified before the adenylin or alpha-sarcoglycan gene. So that makes it a bit more complicated. And this is why the sarcoglycans in particular have always been a bit confusing, because, you know, we call them alpha, beta, gamma, delta, but the classification was, you know, C for gamma and D for alpha, and it didn't really make much sense. Nevertheless, this was the suggestion, and then over, you know, the next more than 20 years, more were discovered. And one protein complex that played an important role was the dystrophin glycoprotein complex, because this is where the sarcoglycans were, and where other limb-girdle muscular dystrophies, including actually caveolin, were associated with this complex. And this was also the complex with a lot of attention to dystroglycan, and many of the limb-girdle muscular dystrophies are associated with that complex. So in 2016, as I said, you know, more than 20 years later, after the first introduction of this classification system, the lab from Carmen Paradas published LGMD2Z with mutations in the POLGLUT1 gene. So, you know, it took about a bit more than 20 years to have this table that you see here. And I think, you know, general lessons were that the dominant limb-girdle muscular dystrophies are much rarer, only 10% of all limb-girdle muscular dystrophies. And the recessive ones were the ones that were most common. And those were the ones also with typical muscular dystrophy features and a high CK. Now, I think many of us who work in muscular dystrophies never really felt entirely, you know, comfortable and confident about this classification, because there were some genes in there, and I think, you know, most prominently maybe the one for Pompe disease, the GAA gene, where you thought, yes, I mean, they present maybe with limb-girdle weakness and have a high CK, but it's not really a limb-girdle muscular dystrophy because it's known as a lysosomal storage disease and metabolic myopathy. So there were duplications of diseases in several classification systems. I just have 3 slides to give you an idea about frequencies as well. And I'm just using our project that is familiar to many of you, the MyoSeq project, where we did exome sequencing in 2,000 patients with limb-girdle weakness. Those patients were over 10 years, and they were supposed to have an elevated serum CK activity. I'm not going into the details, but we had recruited patients from about 50 centers in Europe. This was supported by patient organizations, by industry, and the Broad Institute. And I'm just showing you this to give you an idea, and this was actually not the final summary, but it doesn't really matter. I mean, even after 2,000 patients, the proportions and the ratio hasn't really changed. But what we saw is that the most common forms of limb-girdle muscular dystrophy, clearly, and I think this is something applicable to the global prevalence of limb-girdle muscular dystrophies as well, are the ones caused by mutations in the calpain-3 and the dysferlin gene. There are some countries that have reported more dysferlin patients, but I think generally, globally, calpain is probably the most common one. And of course, there are regions and countries where you have founder mutations where this doesn't apply. And then there are 2 other ones, the one that is caused by mutations in the anoctamin 5 gene, and also the one caused by mutations in the titin gene, that are fairly common. Again, depending on founder mutations, they might be more frequent in other countries. If you then take all those together that cause deficiency of or a hypoglycosylation of alpha-dystroglycan, it's actually also a very big group. It would actually be the 3rd biggest group. And what was interesting in this case for MyoSeq, that there were quite a lot of POMT2 patients, something that we weren't really aware of before. But what's also really interesting is that there are other limb-girdle muscular dystrophies like delta-sarcoglycan here that are extremely rare. So in like 2,000 patients with limb-girdle weakness, we didn't sequence a single delta-sarcoglycan. So some are ultra rare of these diseases, and some are more common actually across most countries. What's also interesting when you talk about limb-girdle muscular dystrophies is that no one has dared so far to call an X-linked disease a limb-girdle muscular dystrophy, despite the fact that clinically, Duchenne and Becker muscular dystrophy are, you would say, the most common forms of limb-girdle muscular dystrophy. They have, shoulder and pelvic girdle weakness and have a muscular dystrophy, but of course had been named according to the people that first described them. So I always say that when you have a male with limb-girdle weakness and a dystrophy, make sure that you first exclude Duchenne and Becker. But the same is actually true for female carriers for mutations in the Duchenne gene or the dystrophin gene. It's more frequent that you have manifesting carriers of Duchenne and Becker than some of the rare forms of limb-girdle muscular dystrophy. So the phenotypic spectrum of these diseases is extremely broad. And despite the fact that they do share limb-girdle weakness, they can also have truncal and distal weakness, and they also share the autosomal mode of inheritance. But I think there would be X-linked diseases that you could, beside Becker and Duchenne, also call a limb-girdle muscular dystrophy. Some patients with FHL1 mutations, for example. Anyway, so it's not a homogeneous group, which makes classification a bit more difficult. And so this was the situation that I've shown you before. And this is when, quite a number of people started to really think, well, what about the next one? What comes after LGMD2Z? And we were actually working on genes where you would say, well, that's a limb-girdle muscular dystrophy gene. And so it's important to understand that OMIM would by default have started with LGMD2AA again. And there were, many colleagues in our community, including myself, who thought, well, we don't want to start with LGMD2AA. That's not very systematic and doesn't really make much sense. Interestingly, there was even a survey by colleagues in France who also felt, well, we need to change something about this. And this is further complicated by the fact that they don't just form a differential diagnosis among themselves, but of course there are many other diseases with limb-girdle weakness. And if you take biopsies, many of these other diseases might even show dystrophic features. So therefore, we submitted a proposal for another ENMC workshop. And so, after more than 20 years, I think the time was there to say, look, we need to address this. And again, there were some people who also joined the workshop in 1995. You see Andoni here, and there's Marian. I should point out that we also had invited representation from OMIM, so Ada Hanosh is here, and then we also had representation from Orphanet, who have their own classification system, and this was Ségolène Aimé, and Marianne also worked on the ICD classification. So we had experts working with other classification systems, and we also had patients there from a number of diseases and patient advocacy groups. Because if you want to change something, you need to make sure that you also listen to the patient voice. Now, many of you will have attended ENMC workshops. You're basically locked into a hotel room for 2.5 days and need to come up ultimately with a report. And so the first day, or first 1.5 days almost, we just spent discussing what is actually defining a limb-girdle muscular dystrophy. And this is the definition that we came up with. We said limb-girdle muscular dystrophy is a genetically inherited condition that primarily affects skeletal muscle, leading to progressive, predominantly proximal muscle weakness at presentation, caused by a loss of muscle fibers. It's fairly generic, and we then specified this a bit more. And this is the only text-heavy slide I think I have. To be considered a form of limb-girdle muscular dystrophy, the condition must be described in at least 2 unrelated families with affected individuals achieving independent walking. So this was something new, and we wanted to distinguish them from the congenital muscular dystrophies. Must have an elevated serum creatine kinase activity, must demonstrate degenerative changes on muscle imaging, and this is mainly muscle MRI, but also other imaging modalities, over the course of the disease, and have dystrophic changes on muscle histology, ultimately leading to end-stage pathology for the most affected muscle. So when you do an MRI, you see that some of the muscles in limb-girdle muscular dystrophy at late stages are completely replaced by fat. That's what we said is typical for these group of diseases. And then we used this definition and said, okay, let's apply this to the classification system. And I'll go through this with you now, and so that you can see what the process was. So first of all, we looked at the dominant diseases. And we said, well, you know, myotillin is normally a myofibrillar myopathy. And many of these patients don't really present with high CKs. They're better known as myofibrillar myopathies. So let's actually not make it too complicated and have the myofibrillar myopathies mixed up with limb-girdle muscular dystrophy. So we thought, let's take that away. And lamin AC, yes, I mean, patients can also present with more of a limb-girdle muscular dystrophy type presentation or features. But what's most important is that they are recognized as Emery-Dreifuss muscular dystrophy because of the cardiac involvement. And clinicians who aren't experts would always associate Emery-Dreifuss with cardiac involvement. So we said that should be, and most patients actually present with Emery-Dreifuss muscular dystrophy features, should be an Emery-Dreifuss muscular dystrophy. And then caveolin 3, I mean, yes, a few patients had been published to present like a limb-girdle muscular dystrophy, but most patients actually show rippling muscle disease and myalgia, and that is a clinical feature that you want to look at. And rippling muscle disease is well known, so you know, let's not list them as a limb-girdle, but rather, you know, rippling muscle disease. And then because we already excluded myotillin as a myofibrillar myopathy, of course, looking at desmin, you would say, well, that's of course a myofibrillar myopathy, the most common one, also known as desminopathy or desmin-related myopathy. So it's more confusing to have it as a limb-girdle muscular dystrophy as well. And then for this locus and this classification, there wasn't even a gene known. So we said, well, that's not really according to our definition, because we said you need to have 2 independent families. You know, you need to have a gene. With those remaining 3, we had discussions and we felt, well, these patients can actually present as limb-girdle muscular dystrophies. And you can argue about this, and we did this, whether DNA JB6 could also, you know, be classified as a myofibrillar myopathy. But clearly there are patients in the literature, and there were some also strong opinions that this is rather, can be justified to be listed as a limb-girdle muscular dystrophy. So let's look at the recessive ones now. Well, I already showed you that these are the traditional ones here. And then talethonin, although very rare, clearly, I mean, you couldn't have a purer limb-girdle muscular dystrophy. But going through this list, and I had already mentioned, you know, Pompe disease, and we had already deleted desmin from the dominant one. So we said, well, let's get rid of desmin here, and let's also get rid of, Pompe disease. And then for PINCH2, BVES, and TOR1-AIP1, there had only been single families reported. And we said, well, you know, you can't be certain that these are for sure limb-girdle muscular dystrophies. And for example, for PINCH2, no other family has ever been reported. We didn't find any in our 2000 patients in MyoSeq. And other big sequencing projects for limb-girdle weakness haven't really identified any PINCH2, for example. So we took them out. And so the remaining ones, everyone felt, yes, these are limb-girdle muscular dystrophies because they're, showing the dystroglycanopathies, and then the sarcoglycanopathies, calpain, dysferlin, telethonin, ANO5, I've mentioned that before, titin. So we said, okay, these are now our clearly agreed limb-girdle muscular dystrophies. And we don't want to run this risk about the end of the alphabet again. So let's list the recessive ones with an R and the dominant ones with a D. And then we can have numbers in order of their characterization. And it is actually a system that Omen has applied to other rare diseases. So when we cleaned up our classification, this is how it looked. So these were all the ones that we had discussed that were previously listed as limb-girdle muscular dystrophy. So we took some out, but then we also had a discussion, should there be some diseases that we would add to the list? And so these are now the genes that we added. And I'll again explain why. So for collagen, for example, we have Ullrich congenital muscular dystrophy. It's defined as a muscular dystrophy. But the onset is very early, and there are, of course, additional features. There are many Bethlehem patients. And here in Newcastle, we run the reference center for limb-girdle muscular dystrophy patients in the UK. And we often saw that patients were referred to our service that turned out to be Bethlehem myopathy, but actually had high CK levels, had dystrophic features on the biopsy. They also had the imaging characteristics that we defined. So we felt that collagen 6 related disorders should also be listed under the limb-girdle muscular dystrophies, because a neurologist who would see a patient with Bethlehem myopathy might rather think about limb-girdle muscular dystrophy than Bethlehem myopathy. And then clearly over the past 25 years since the laminin alpha-2 gene had first been reported to be the most common gene affected in congenital muscular dystrophies, there have been many milder patients that can also show a classical, typical limb-girdle muscular dystrophy phenotype. So we felt that this is really important to add. For collagen, we knew that there were both recessive and dominant modes of inheritance, whereas for laminin, only recessive ones. And then there are a few reports about limb-girdle muscular dystrophy patients with dominant calpain 3. So we thought, well, it's important to at least think that calpain 3 patients can also present in a dominant mode of inheritance. There's still some debate around this, and it's clearly a minority, but nevertheless. So there was an agreement that this is what we use, and this is what we published. This was a new classification. I want to point out that it has been really helpful that we had patients at the workshop, and that Madelon Kronemann, who is also actively involved, of course, in EURO-NMD and in the muscle group, she attended the NMC workshop, and together with Marianne also then worked on a patient information leaflet on those new names, because this had quite a lot of implications for patients, particularly for those who were labeled limb-girdle before, and we felt, well, actually there should rather be Emery-Dreifuss or myofibrillar myopathy or Pompe disease. And I had to learn as well that what is really important is to have broad stakeholder discussions, because there were colleagues that weren't happy with this. They didn't participate in the workshop, so didn't follow all the discussions we had, because we didn't make any easy decisions. But I would probably for the future say, well, you need to reach out even further and have broader discussions. I'll give you one example. When I visited Japan, I learned that there were colleagues working on an exoskeleton for limb-girdle muscular dystrophy. And they had approval for this, and they could also do their research in patients with Pompe disease because they were classified as limb-girdle muscular dystrophy. When we published our paper and took Pompe out, it affected their research because all of a sudden they weren't allowed anymore to study the exoskeleton in Pompe patients because they were only allowed to do it in limb-girdle patients. So you realize that there are ramifications and implications if you change classifications. So, and it wasn't just that, well, this is now the complete list. Once we published this, there were also colleagues who said, wait a minute, I mean, you know, we published on a disease that we call limb-girdle muscular dystrophy, but you didn't list it. One of them was, for example, in POMGNT2, and Ichizu Nishino's group had published on this. And yes, after looking at those patients, it indeed looked as if they have clear limb-girdle muscular dystrophy. So they have now been added. They're not within our publication initially from the ENMC report, but OMIM now classifies them as, in this case, LGMD R24. And it's also that we have worked on two other genes, and one you have seen before, BVAS, formerly called also POPDC1. We had taken it out because there was only one family that was reported. But meanwhile, quite a number of families have been reported with clear limb-girdle muscular dystrophy features, as summarized in our definition. So LGMD R25, BVES related is back on the list. And then a very similar gene, POPDC3, had been published also with limb-girdle muscular dystrophy, and that's on the list. To my knowledge, this is the current classification that is also listed on OMIM. But, and I think, again, this is important to understand, several of these genes can, of course, also cause other diseases. And they can, for example, cause congenital muscular dystrophies. And OMIM also lists dystroglycanopathies as a separate disease group. So several of those are also listed under the dystroglycanopathies. So in the last few slides, I want to also then point out that there are other initiatives that look at a classification system. Now, many of you might be part of clinical genome resource groups that look at the annotation of genes for certain muscle diseases, ClinGen. And so I'm part of the Limb-Girdle group, and I have to say I was really impressed by the approach, which is different from the approach that we took at the ENMC workshop. Because here, the overall goal is to build a knowledge base that defines the clinical relevance of genes and variants for use in precision medicine and research. So the approach here is not from the clinical side really, but very much from the gene and variant side. Which genes and variants can cause a limb-girdle muscular dystrophy? And so ClinGen has established a system here where they look at the evidence that a genetic alteration of a gene causes the disease in question. And in order to rate the evidence, they've come up with a semi-quantitative framework to assess the strength of evidence supporting a gene-disease association. From, yes, this is definitely causing, for example, a limb-girdle muscular dystrophy, or, you know, the evidence is only moderate or limited, or, you know, is actually there's very little evidence. And so these are, you know, defines criteria need to demonstrate causality. These are some of the criteria. Organizes and quantifies different types of evidence, allows curators to methodologically assign a clinical validity classification. So there's a classification that you use. I would advise people to maybe go onto the ClinGen website if you want to learn more about this. There are also very good publications about the system that is applied. Now, of course, you nevertheless need to have some working definition of the disease group. And I have reduced this to these 3 bullet points. This is a bit the working definition of LGMD that ClinGen is currently using. So progressive weakness in limb musculature, pelvic and shoulder girdle, independent ambulation by the age of 2 years. So they have been a bit more specific. We just said independent walking. Elevated CK of, over 2 times the upper limit of the norm. And a muscle biopsy with dystrophic and myopathic features. So we are still discussing this. We haven't defined yet really whether ClinGen's definition of a limb-girdle muscular dystrophy will be different or just more elaborate than the one from the ENMC. And there are 2 working groups, one that is doing the gene curation expert panel and one the variant curation. So the gene curation expert panel is basically looking at whether the genes that are currently assigned to limb-girdle muscular dystrophy are justified to be called limb-girdle muscular dystrophy. And then the bigger job is to actually look into mutations, because some mutations in those genes may never cause a limb-girdle muscular dystrophy, but will always cause a different phenotype. For example, in some of the dystroglycanopathies, certain mutations will cause Walker-Warburg syndrome or muscle-eye brain disease, but never a limb-girdle muscular dystrophy. So you still need to look at the variants as well. And then you need to look at the evidence. How strong is the evidence? So it's a lot of work, and this isn't just one workshop, but these are continuous workshops with continuous discussions looking at individual genes and individual variants. And so for the limb-girdle muscular dystrophy groups, Chris Weil from St. Louis, Washington University there is the chairperson. We have so far curated 16 genes. 3 are currently in progress and 19 genes remain to be curated. As I say, the process is a lot more thorough than what we did at the ENMC workshop. And for the variants, it's even more complex, as you can imagine. And the approach, therefore, was that we will primarily look at 7 autosomal recessive limb-girdle muscular dystrophy genes, because these are the most common ones and the ones where we definitely 100% know that these are limb-girdle muscular dystrophies. And the reference is the Academy of Clinical Medical Genetics guidelines. And so, yeah, this is an ongoing process. I just want to give you an example that really impressed me about this ClinGen approach. So one of the genes that we were, for example, looking at was the POMGNT1 gene. Now, the POMGNT1 gene had been published to be associated with limb-girdle muscular dystrophy patients. But when within the ClinGen working groups, we thoroughly looked at this, it turned out that the mutations, or the claimed mutations, were actually polymorphisms. And so none of the publications fulfilled the evidence criteria that POMGNT1 is indeed a limb-girdle muscular dystrophy gene. Now, meanwhile, I have been in touch with colleagues, Vincenzo Nigro and Bjarne Ut, and Maturi Hedge in the States, and Kevin Campbell as well. And we have looked at sequences, exome sequences in particular, of more than 10,000 patients with limb-girdle weakness. And not a single patient was identified with POMGNT1 gene mutations. So maybe, we don't know yet, this is either extremely rare or mutations in POMGNT1 never cause a limb-girdle muscular dystrophy and only cause muscle-eye brain disease or congenital muscular dystrophy or Walker-Warburg syndrome. So it was really interesting to go through this process. So at the ENMC workshop, we didn't really go through these, variants that had been published in detail, but maybe we need to take this out. Another gene that just illustrates the process is actually the one that we published, the POPDC3 gene. So, John Fissing was the first author of this paper. We had 3 families. They all really showed limb-girdle muscular dystrophy. The MRI also met the criteria. The biopsy, some were a bit more myopathic, but others were really dystrophic. We were fairly convinced that, yes, this is a limb-girdle muscular dystrophy. The CKs were, 2,000, 9,000 here, 7,000. Interestingly, all of these patients had missense mutations. And, Jackie Beckman, rightly pointed out, well, this isn't enough for a lot score of over 3. So, whether this is really the gene, because we didn't do any linkage analysis. And then all of this is just modeling. And in terms of modeling, Klingen says that isn't hard evidence. So even though we could show that this is in these missense mutations are affecting preserved areas of the gene, they also affect the relevant catalytic domain and reduce binding for CAMP, one important function of POPDC3. We also show that a knockout in zebrafish causes a phenotype, but those weren't missense mutations. So overall, and we did some functional assays in TRAC1 channels as well in xenopus oocytes. Nevertheless, if you apply the strict criteria from Klingen, there wasn't much evidence that this is indeed a limb-girdle muscular dystrophy. So it depends a bit on the evidence criteria you apply. So I think what is important, and this is my last slide now, if we think about the classification, the one that we came up with at the ENMC workshop will most likely not be the ultimate classification. And I've already shown you that for POMGNT1, currently we don't have any evidence that it's a limb-girdle muscular dystrophy. Nevertheless, we published it as a limb-girdle muscular dystrophy because we thought that there were actually 3 publications that were okay. And only when you go through this with a fine-tooth comb, you realize maybe not. But it also illustrated that there are various classification efforts now across neuromuscular diseases. Klingen also has groups for the congenital myopathies and congenital muscular dystrophies, for example. So we need to make sure that we harmonize our classification and definition approach across these different efforts. And of course, there's now, the ICD-11 classification that people are working on, and they particularly consider rare diseases. So we need to also make sure that we harmonize this with international efforts around rare diseases in general. But we also need to ask ourselves the question, well, for whom are these classifications? Ultimately, as a clinician, I think they should be based on clinical phenotypes, that the non-neuromuscular expert sees a patient with limb-girdle weakness and say, in a high CK, and says, well, this is a limb-girdle muscular dystrophy. And you can't just come probably from a variant perspective. So my own lessons that I have learned after changing the classification is you need broad consensus. And this is something that is currently ongoing. So we are discussing across several of the Klingen groups. Now maybe a new harmonized classification system. These efforts are supported by the World Muscle Society. We have had discussion already with industry as well and with patient communities. Because this is becoming more important now also that we have new therapies, what is the indication of your drug? So if you approve a drug in a limb-girdle muscular dystrophy, can you also treat patients that have mutations in the same gene that have congenital muscular dystrophy? So industry wants to have some clarity about this as well. Our classification system is fairly messed up. And so, to make sure that as many patients as possible get access to to medicines that are effective and safe with a certain indication, we need to make sure that the classification system doesn't hamper that approach. So I would like to thank my colleagues again that joined the ENMC workshop. And I would also like to thank all experts and biocurators from the ClinGen LGMD expert panel. And thank you very much for your attention.
Thank you, Volker. I think this was a wonderful presentation, and that it gives lots of food for thought. I liked indeed about the historical background that you started with, and basically it reminds me of the problems of the philosophy of reality. So if things exist by themselves and you can describe them, or if they don't exist by themselves. So you did initially a very good description of all the phenomenology behind the disease, and then trying to go to another paradigm to explain the occurrence. So we have three questions. The first person to question you was Marianne de Visser. You can unmute yourself, Marianne, please, and voice the question.
Yeah, first I would like to thank Volker for an excellent lecture, and also the neuromuscular community is very grateful that you had the vision and also the courage to start this initiative to discuss the classification system of limb-girdle muscular dystrophies, because I was involved a few years before the ENMC workshop in a similar exercise on Charcot-Marie-Tooth disease. And there has not been a workshop on that, only a survey, but I can tell you that that has still not materialized, unfortunately. So we are stuck there with the old classification system. So thank you very much for that. I had a question about the ClinGen definition. In the ENMC definition, there was the possibility to choose between muscle biopsy features and MRI features, whereas in ClinGen, there are only muscle biopsy features are part of the definition, but currently the clinicians don't do a muscle biopsy anymore when they first see a patient with proximal muscle weakness and a high CK.
Yeah, Marianne, so first of all, thank you very much for the kind words. And this shows one of the challenges. And so classification systems are also depending on cultural or geographic or country-specific diagnostic approaches. In the US, MRI is not a common tool to assess muscle patients, and it boils down to costs. And it's just the insurance system in the US that is different from the way we do imaging here in Europe. So because ClinGen was a US-based initiative, they hadn't really thought about MRI. But you can imagine that over the past year, or it's probably going on for longer, 2 years, more and more we realize how important MRI can be. And I'm fairly confident that when we finalize a LGMD definition also through ClinGen, that imaging will be mentioned.
Thank you.
Excellent. So we have a question from Joseph Camis. Do you want to unmute yourself, please?
I prefer you read the question, my question.
Okay, no worries. So many patients with Limb-Girdle syndrome are biopsied on more than one occasion throughout their lives, distrusting the lab pathology or the selection on biopsied muscle. In your opinion, what role does this invasive procedure currently play?
So this goes back a bit to what Marianne de Visser has just pointed out. The imaging is often now starting to be the first investigation before you go into more invasive ones. I think the the biopsy still plays a role for limb-girdle muscular dystrophies, but only if you do a comprehensive immunoanalysis. Just for histology, it often is fairly meaningless. Limb-girdle muscular dystrophies can show a broad spectrum of histological features. They don't even always need to be dystrophic, but immunohistochemistry can help in immunoblotting. So I do still think that there's an important role for muscle biopsy analysis, but I would always do an MRI now before, for two reasons. One, you can decide which muscle to biopsy. And the second, more important one, you might even make a diagnosis based on your clinical phenotype and the MRI and don't need a muscle biopsy.
So there is an anonymous attendee that asks if there are LGMDR in connection with DPM3 and JAK2 and myofascin.
Yes, so some of those genes that you've just mentioned, JAK2 has only been now described, and I think that authors sometimes are a bit hesitant to classify their disease. They rather describe everything, and then it is OMIM, for example, that might assign the disease to a specific group of, for example, limb-girdle muscular dystrophies or congenital muscular dystrophies. My personal opinion is that JAK2 would be a new limb-girdle muscular dystrophy gene, a recessive one. So let's see. I mean, it has only recently been published. What OMIM does, whether it would assign, for example, R27 to JAK2. For DPM3, I would agree that some of these patients form an important differential diagnosis to limb-girdle muscular dystrophies. We have followed up patients for years and thought this is a limb-girdle muscular dystrophy before we then ultimately found the gene. So yes, I think that the classification as it stands now isn't complete, and you will find patients with limb-girdle muscular dystrophies that meet the definition with mutations in a number of metabolic myopathy genes, for example.
Another question from Muravi Rayani is about the role of ultrasound in limb-girdle muscular dystrophy, if there is any role or not.
I think that the role of ultrasound is becoming more important, but probably applies more to infants and to early childhood, rather than the diagnostics of adult onset, and most limb-girdle muscular dystrophies are, of course, of later onset. So for limb-girdle muscular dystrophies, I would still think that muscle MRI is more meaningful, which is different for the smaller your patient is. So I don't think that the ultrasound is that relevant for limb-girdle muscular dystrophies.
So Professor Toscano, Antonio Toscano, has raised his hand. I don't know if he has a question.
Hi, everybody. Thanks. No, the question was very simple. And Volker has already written the response. The question was, how many families with the POPDC3 have been recognized? And my curiosity is if they come from the same geographic area.
So they come from 3 different countries, so are not related. There's 3 families, all of which show similar features, high CK, clear limb-girdle weakness, and on MRI, fairly similar features. So if you want to look at the publication, it's in Annals of Neurology, and first author is John Fissing. But those are the only ones that I'm aware of that have been published.
Okay. So Michael Rataj, do you want to unmute yourself? and put your question. Do you want me to read
No,
it?
maybe sorry, Antonio. Maybe you can read it from the chat.
Okay. So is there currently available any genetic test for which type of test you can recommend, which will be able to define clearly what type of limb-girdle muscle dystrophy an adult patient has?
So, I mean, the basis for the diagnosis is, of course, the clinical assessment and the family history. And many centers are now going to do either gene panels for limb-girdles or exomes, or, you know, in the UK, we are now going to genomes. This is why this ClinVar approach is so important. I think for certain limb-girdle muscular dystrophy, it's easy. If you have a patient with limb-girdle muscular dystrophy and elevated CK and 2 nonsense mutations in calpain or 2 missense mutations, that's probably your gene. So there's no straightforward answer to address all limb-girdle muscular dystrophies in your question. But what I would currently recommend is you do the medical assessment, the family history, you do a CK, and then you do genetic testing. If you have access to a large panel, that's what I would do.
Excellent. Now I want to provoke with a question, if you allow me. And my current boss, you used to be my boss in the past, as you know, but perhaps some of the people present don't know. But my current boss was on the attendance. I don't see her anymore. She's there. She's L Bon. So on behalf of her, I will challenge you with the lamin AC gene. So the question I wanted to ask you, and it's not to you, of course, it's to the current efforts of classification, is about the clean gen approach. So in the case of lamin AC, you can go to the variant level and still have 2 different phenotypes. So how can you rely basically on defining gene and variants when at least for that gene, you still can have different diseases arising from the same mutation?
So the challenge, Antonio, you're absolutely right. That is a challenge. And this is exactly why an approach like the ClinVar, variant curation and annotation approach is so important, because you're absolutely right. As a clinician, if you get a variant, you ideally want to know, well, what does this mean for the patient? And yes, there are some variants, and I mentioned, some of the dystroglycanopathy genes. FKRP is a good example as well. If you have a nonsense mutation in FKRP, it will never be a limb-girdle muscular dystrophy. It will always be something a lot more severe. So yeah, we need to pay attention. This is the complexity that we are living in as experts dealing with rare genetic diseases, that it boils down to, you have to know your genes, you have to know your phenotypes, and ideally you have to know your proteins as well. So it's too easy to just point to, oh, there's a mutation in that gene and this is what it means. Yeah. Just maybe one more comment. I mean, what we start seeing now is also that for more and more genes, we see that there's recessive and dominant mode of inheritance. So even our classical approach that, oh, these are the recessive ones and these are the dominant ones, I think is something that is becoming more blurred.
Excellent. I think that Giselle is sending a kind of question through the chat saying that any classification needs to be revised. So do you know about plans about revision of this classification?
Yes, I mean, that's in my closing remarks. I did mention that we are now talking across several consortia. What I can envisage is that we will have actually a larger conference, supported also by the World Muscle Society, to discuss a more harmonized classification system. And Marianne de Visser mentioned the neuropathies. how challenging it is for them. Nevertheless, the neuropathies have been a lot more systematic than the muscle diseases, because they started from neuropathy, myelinating, demyelinating, and then went to the modes of inheritance. Whereas in muscle, we have, sometimes named muscle diseases after the people that first described them, the muscles that are most severely affected, associated symptoms, onset of the disease. So it's very confusing, often just based on histology and histological features. So we don't have a systematic approach to genetic muscle diseases when it comes to nomenclature. And I agree with Giselle, you probably need to constantly revise this based on the knowledge that you have.
Excellent. So we have a lull in the question string. So I think that I will start by thanking you very much, Volker, again for this very nice and very educative webinar. And we'll have certainly many other occasions in the future to discuss with you about this and other subjects, of course. And I think it was a pleasure for everybody present here and for everybody that will watch this webinar to go through this matter that, as you say, is complex, but it's rather important for our patients. Thank you.
Thank you very much, Antonio. And thanks to everyone for listening.
Thank you. Bye now.
Bye.
Thank you. Bye-bye.